So with this presentation, we introduce an example for an integrated approach to testing mixtures and their aerosols in vitro. We present results from our studies applying a tiered systems toxicology approach to gain insights into the potential toxicity of the test item and the underlying mechanisms. Systems toxicology is an integration of systems biology and toxicology. It combines comprehensive molecular analysis with quantitative modeling to understand the characteristic of a biological system as a whole. Using systems toxicology here at Philip Morris International, we aim to understand the detailed mechanistic biological processes that occur following exposure, not only to cigarette smoke, but also to aerosols of potential modified risk tobacco products. Previously, we have proposed a framework for in vitro systems toxicology assessment of e-liquids and their aerosols, intended to actually complement the battery of standard classical genotoxicity assays. The framework outlines a top-down characterization of e-cigarette liquids and their aerosols, starting with chemical characterization of the e-liquids and toxicity studies using simple cell systems. This step would then lead to a selection of low toxicity e-liquids, which are further tested using mechanisms of toxicity-specific readouts, with, for example, high-content screening or HCS, also still in simple cellular systems. E-liquids meeting predefined toxicity criteria are then aerosolized and tested using three-dimensional organotypic models for a detailed systems-level analysis of cellular mechanisms of toxicity. This framework really has shown utility in our hands, and so we believe that it can be applied to testing other mixtures and compounds with suspected respiratory toxicity. And so here we will present case studies applying the proposed framework for the in vitro assessment of cigarette smoke and aerosol from a potential modified risk tobacco product, what we refer to as the tobacco heating system, or THS 2.2. The first layer of the proposed framework aims to screen the general toxicity or cell viability of various formulation or other inhaled compounds efficiently, independent of the aerosol generation devices. We recommend the use of relevant in vitro test systems. For example, in the context of inhaled compounds, cells of the respiratory system would be relevant. For a relatively inexpensive rapid screening, two-dimensional monolayer culture systems are appropriate. These monolayer two-dimensional culture systems are, of course, easy to handle. They're relatively inexpensive and suitable for large-scale studies. Multi-parametric indicators of general toxicity can benefit from real-time cellular analysis and HCS assays. Recent technologies such as the Inhalation platform enable measurement of cell viability in real time, allowing a simultaneous monitoring of the time-dependent exposure response. Alternatively, high-content approaches based on continuous multiplex readouts can be used to monitor multiple markers of cell death simultaneously on a cell-by-cell basis. The different probes can be employed to discriminate viable cells from non-viable cells and apoptotic cells from necrotic cells. This ability to distinguish between the aspect of cell death is also clinically relevant, particularly in developing specific therapeutic compounds targeting different processes of cell death. Here we present a use case in which the Inhalation platform is used to monitor cell viability following treatment with different cigarette smoke and heated tobacco aerosol fractions. Cell viability is seen on the y-axis here, was measured as a change in electrical impedance over a 4 or 24-hour exposure to the aqueous extract, to the gas vapor phase, or to total particulate matter from the smoke of 3-alpha-F standard reference cigarettes, which we obtained from the University of Kentucky, and from the aerosol of the THS 2.2. The red line here indicates cell viability of cells exposed to 3-alpha-F cigarette smoke fraction, and the purple line indicates cell viability of cells exposed to THS 2.2 aerosol fractions. The green horizontal dotted line indicates the 50% cell viability. The data show that exposure to all three 3-alpha-F cigarette smoke fractions actually cause dose and time-dependent decreases in cell viability. This effect we can already see with higher concentration at 4 hours, but it was also more evident across most concentrations at 24 hours. On the other hand, THS 2.2 gas vapor phase did not have any effect on cell viability independent of time, whereas exposure to THS 2.2 aqueous extract at high concentration for 24 hours and THS 2.2 total particulate matter at high concentrations for both 4 and 24 hours resulted in a small decrease in cell viability. The information obtained from the first layer assessment can be leveraged to determine the selective compounds or formulation that should be tested further in the second layer assessment. So for example, one may further select the compounds that were shown to elicit considerable reduction in cell viability. The second layer aims to identify the toxicity mechanisms of the tested formulations beyond the impact of cell viability. This table here actually shows a summary overview of endpoints that are included in our routine toxicity profiling of various compounds of interest using an HCS platform. Markers such as the mitochondrial health, DNA damage, and oxidative stress are most commonly investigated. For this type of studies, we routinely use in vitro human lung and cardiovascular models, such as normal human bronchial epithelial cells and human coronary aortic endothelial cells grown in monolayers. So for example, we use high-content screening to further investigate the effect of exposure to cigarette smoke and heated tobacco aerosol aqueous extract on oxidative stress and DNA damage. Oxidative stress was monitored by measuring the formation of reactive oxygen species, or ROS, and glutathione, or GHH, content following a 4 or 24-hour exposure. We showed here the values normalized to vehicle control, which was actually phosphate buffercellin treated cells, for which the relative unit was set into 1. So we noted an increase in ROS formation following treatment with high doses of 3-alpha-F aqueous extract for 24 hours, but not for 4 hours. Because this was actually accompanied by cell loss, as we have seen in the previous slide under the first layer assessment, we believe it was not likely to reflect through oxidative stress. There was no indication of ROS formation in cells exposed to THS 2.2 aerosol aqueous extract independent of treatment duration. However, extracts derived from both items cause a dose-dependent decreases in GHH content at both time points, with noticeably larger decreases in the 3-alpha-F treated cells than the THS 2.2 extract treated cells at comparable doses. The genotoxic potential of the aqueous extract from THS 2.2 aerosol relative to that of 3-alpha-F cigarette smoke was then evaluated by immunostaining of cells for gamma H2AX as an indicator of DNA double-strand breaks. Exposure to 3R for FAQS extract at the two highest doses here resulted in an increase in gamma H2AX signals, while exposure to DHS 2.2 aqueous extract actually did not. Finally, the third layer of the framework aims to evaluate the toxicity-related mechanisms of the aerosols generated from selected compounds or formulations. Various aerosol generation devices are available, as we can see here, such as the condensation monodispersed aerosol, the vitro cell cloud system, and a simple nebulizer. For electronic cigarettes, actually the aerosols can be generated manually using a syringe and a timer, or using a semi-automated system consisting of a vacuum pump and flow controllers, and also using smoke machines that is adapted to aerosolize electronic cigarettes. A suitable exposure system is also needed to conduct a proper exposure study. The vitro cell cloud system is specifically designed for the controlled and spatially uniform deposition of liquid aerosol on cells cultured at the air-liquid interface. In our laboratory, we actually use the VitroCell 2448 exposure system. The system is equipped with a dilution and distribution module that is used to control the concentration of aerosol fed into the exposure chamber, and with a cultivation-based module that houses the culture during the exposure experiment. The characteristic of the generated aerosol, for example, the particle size, distribution, and composition, vary depending on the aerosol generation device. The physical configuration of the exposure system or chamber will also impact the deposition of the test aerosol in the biological test system. So for this, the characterization of the test aerosol should also be conducted as part of the assessment. Organotypic air-liquid interface in vitro models have been increasingly used in research and also in toxicological studies. They can be certainly developed using primary human cells, thus allowing for investigation into mechanisms that occur in humans, some of which might actually differ or do not exist in animal models. Morphologically, these models mimic their tissue counterparts better than classic submerged in vitro models. Also, the air-liquid interface setup allows for direct exposure to inhaled compounds, providing a physiologically relevant test system. Considering that multiple studies reported cigarette smoke exposure eliciting tissue injury throughout the aerodigestive tract, cultures derived not only from the lung, such as bronchial or small airway epithelial cultures, but also from oral and nasal tissues are going to be relevant for the assessment of cigarette smoke and electronic cigarette aerosol. We, of course, can evaluate various endpoints in the culture medium or tissues. So, for example, histological examination of the tissue section can be conducted to assess the effect of exposure on culture morphology. Messenger RNA and microRNA expression can be assessed in tissue samples, whereas different soluble mediators can also be measured in the culture medium. And for ciliated cultures, we can obtain video recording to monitor cilia beating frequency. And finally, the choice of endpoints will certainly depend on the type of tissue that we will be using, and to have a good understanding of the biological behavior of the various tissues is going to be helpful in selecting appropriate sampling times. We actually applied this third layer of the framework in a series of studies using human organotypic buccal, bronchial, and nasal epithelial cultures. In these studies, we assessed the impact of exposure to heated tobacco aerosol relative to that of 3RF cigarette smoke. We conducted a series of studies following the same experimental design to increase really our confidence in measuring accurate exposure effects. We actually exposed the cultures to the heat diazotobacter aerosol from THS 2.2 and 3-alpha-F cigarette smoke at similar nicotine concentrations. So using the VitroCell 2448 exposure system, we adjusted the concentration of the aerosol and smoke to reach this target nicotine concentration. We present here representative images of the hematoxylin eosin stain section of cultures 2 days after exposure. This work allowed us to examine exposure effects on tissue morphology. For the bronchial and nasal cultures, Gaussian blue staining was actually performed to visualize the presence of goblet cells. The results show that exposure to the THS 2.2 aerosol shown here did not cause morphological alteration in any of the 3 culture types. However, exposure to 3-alpha-F cigarette smoke at similar nicotine concentration caused marked damage in this culture. Next, we profile the expression of various inflammatory mediators in the media. The figure on the left shows the concentration of various mediators that were increased, which are shown in red, or decreased, shown in blue, following exposure relative to the concentration found in cultures exposed to air. The greater changes in the concentration were seen following exposure to cigarette smoke than to THS 2.2 aerosol. We actually also found that the expression of genes profiled in the tissue samples shown here in the x-axis correlated nicely with the expression of the corresponding protein in the medium, as we've shown here in the y-axis. For a part of the system-level investigation, we actually profiled global gene expression in cultures collected at 4 hours, 24 hours, 48, and 72 hours following exposure using DNA microarrays. We found that at similar nicotine concentration, exposure to THS 2.2 aerosol resulted in smaller numbers of significantly differentially expressed genes than exposure to 3-alpha-F cigarette smoke in all 3 cultures. And for us to really obtain meaningful biological interpretation of the complex transcriptomic data set, we used the network enrichment analysis approach. This analysis uses transcriptome data as an input and utilizes causal biological network models and a computational algorithm to infer really the impact of exposure on various biological processes or pathways. The algorithm is known as the Network Perturbation Amplitude, or the NPA. This was then used to compute the exposure-induced perturbation score on each node of a model. We've shown here an example of a network model called the xenobiotic metabolism network. The perturbation score of the entire network model was thus used to infer the exposure-induced impact on a certain biological processes or pathways that were actually modeled in the network. And our collection of network models can be explored at the causalbionet.com website. We built the models using biological expression language, or BEL. These BEL-scripted networks describe pathways and processes relevant in disease and non-disease pulmonary and vascular tissues described in literature. There is this belief platform that is actually available for a semi-automated generation of BEL statements from literature statements, and it can be used to create such network models. So using the causal biological network models and the NPA algorithm described before, as well as the transcriptome data from the studies, we found that exposure to THS 2.2 aerosol resulted actually lower impact on various pathways and processes than exposure to 3-alpha-F cigarette smoke. The reduced impact following exposure to THS 2.2 aerosol was observed similarly in all culture types. So in summary, we have shown that various in vitro approaches can be integrated for a relevant assessment of inhaled mixtures. The ultimate goal of monitoring the effects of test compounds on cell viability is to predict in vivo toxicology based on their toxicity profile in vitro. For initial screening, real-time analysis and HCS assays are suitable for relatively rapid screening. But one needs to consider that the characterization of the inhaled aerosol is actually an important part of the assessment because the characteristics of aerosols vary depending on the aerosol generation device. And one should also think of the appropriate biological test system and exposure modes to be used, as well as really the appropriate dosing to better extrapolate human health risks. Nevertheless, we believe that systems biology and omics approaches will increase sensitivity to reveal changes at the cellular and molecular levels otherwise not detected in classical toxicity assays. And finally, a continued collaboration among the scientific community that include industry and regulatory bodies is going to be needed to improve research strategies related to inhaled aerosols and mixtures. So finally, we thank you for your attention. We provided here a list of the relevant publications for your reference and also for additional information. And here is our contact information. So we would be happy to answer any question you may have or to provide perhaps additional information related to this topic. Thank you very much.